Good morning, everyone. Today I'm going to cover the topic of cell physiology, where we are going to cover a number of objectives, and these are as follows. The objectives are, number one is to define physiology and know the difference between physiology, anatomy, and pathophysiology, and then to know the different levels of organization, as we have 6 different levels of organization in the human body, and then to know the physical structure of the human cell and know the function of the different organelles in the human cell. So, as you know, anatomy is the study of body structures and the relationship among them. Physiology is the study of body functions, including the study of a very important concept in physiology, which is homeostasis. And this means keeping the internal environment of the body stable in a changing external environment. And pathophysiology is how changes in the normal physiological processes can lead to disease or injury. Of course, the structure and function are so closely related, so the structure mirrors the function. For example, the thin air sacs or the alveoli of the lung permit the movement of gases from the lungs to the blood, and this function cannot be done by another type of cell that has different structural features. Here we can see the different levels of organization, and we can see here the first level, which is the chemical level, where atoms combine together to form molecules. And so you can see different atoms of different types. They combine to give me different molecules. These molecules in very large numbers can combine to form the different components of a cell, and in this example we see the smooth muscle cell. So the molecules will give me cells, and this is called the cellular level of organization. As you see here, cells are made up of molecules. Then when we combine cells of the same type, so cells of the same type are combined, they form tissue, and this is called the tissue level. In the human body, we have 4 basic types of tissues. We have the nervous tissue, we have the muscle tissue, we have the connective tissue, and we have the epithelial tissue. So any 2 of these basic types of tissues, or at least 2 of these, so 2, 3, or 4 of them, when they are combined together, they can form an organ. So organs are made up of different types of tissues. As you can see here, we have a blood vessel. It is composed of a lining composed of epithelial cells, and this is epithelium. Then we have a muscle coat composed of smooth muscle cells. This is muscle tissue. And then we have an outer coat of connective tissue, and this is composed of fibroblast and collagen fibers and belongs to the connective tissue. Then when we have organs that serve the main function, one main function in the body, these organs will combine together to give me a system, and in this case the cardiovascular system, which is composed of heart and blood vessels. Then when we combine all the organ systems in the body, like cardiovascular, respiratory, alimentary, urinary, skeletal, muscular, and so on, all together they will form the organ system, the full or the whole body of the human being. So the chemical level, atoms are combined to form molecules. Then we have the cellular level, that is where cells are made of different molecules. Then the tissue level, that consists of similar types of cells. As I said, we have 4 basic types of tissues. And then we have, when 2 or more tissues combine together, they form an organ, so made of different types of tissues. Then the organ system, it consists of different organs that work closely together to perform one main function. Like the respiratory system, its main function is to bring oxygen to the blood and get rid of carbon dioxide. the alimentary system. Main function is the digestion and absorption of nutrients that we need them as a source of energy and also as a source of molecules and ions that are necessary for growth and other body functions. Then we have the organism, which is made up of the organ systems. Now let's talk about the cell. This cell is the basic structural and functional unit of the human body and all other living multicellular organisms. It is a highly organized molecular factory, and based on their function, we have a great diversity of function, and we have in our body at least 200 different types of cells. And the organ physiology is derived from the complex functions of these 200 different types of cells that are present in our body. The cell has three main parts, and these are the cell membrane that separates the cell from its exterior or external environment. We have the cytoplasm, which is composed of cytosol. Cytosol is mainly water plus different molecules and ions, and then we have different organelles that have different functions, and I will cover them in the remaining part of the lecture. And not to forget the largest organelle inside the cell, which is the nucleus. As you can see in this slide, this is a cell. We have the cell membrane here in light blue. It is completely separating the cell from its exterior environment, and then we have the large organelle, or the largest organelle in a cell, which is the nucleus, and we have a smaller organelle inside the nucleus called the nucleus, and then we have the different organelles that are present in the cytoplasm. Among these are the mitochondria, the energy factories inside the cell. We have another organelle called the endoplasmic reticulum. As you can see, we have two different types of endoplasmic reticuli. We have one with ribosomes. As you can see here, the dots, the light pink dots. This is a rough endoplasmic reticulum that has ribosomes on its surface, and we have endoplasmic reticulum without ribosomes, and this is the smooth endoplasmic reticulum. We have here this organelle, which is called the Golgi apparatus, which has very important functions in the body that we'll see them later. We have lysosomes, we have peroxisomes, and we have the centrioles among the different organelles that are present inside the cell. In this inbox here, you can see that the cell membrane is composed mainly of a phospholipid bilayer. So you have one molecule of phospholipid and another molecule of phospholipid. They have a special arrangement where each molecule has hydrophilic heads, or each molecule has a hydrophilic head and hydrophobic tail. So if we put many molecules in a solution, they will assume this position where the hydrophobic fatty acid tails face each other, and the hydrophilic phosphate heads will face the water, either on the inside of the cell or the exterior of the cell. So you can see here, this is a phospholipid molecule. It has a hydrophilic head that likes water and hydrophobic tail that hates water. So it tries to stay away from water. It is more of lipid soluble. As I said, if we put thousands of molecules, they will assume this arrangement. This is the arrangement which is the resting arrangement for the phospholipid molecules, where the hydrophilic heads face water on both sides and the hydrophobic tail will face each other. And since we have two layers, we call it the phospholipid bilayer. Bi means two. So now here we can see the portion of the cell membrane, also called the plasma membrane. As you can see, the plasma membrane is not only composed of phospholipid bilayer, but we have also other molecules. We have some cholesterol molecules that we see them here in orange or yellow, dark yellow, and we have those purple huge molecules. We call them membrane proteins. Some of them fully span the phospholipid bilayer from one side to the other side, and we call them integral membrane proteins. Some of them partially span the phospholipid bilayer, so also they are called integral membrane proteins. We have proteins that are attached to the surface of the phospholipid bilayer, mainly on the inside, but sometimes also on the outside of the phospholipid bilayer, and we call them the peripheral membrane proteins. Also, I want to attract your attention that carbohydrate chain might attach to the phospholipid or to the protein. So we have glycolipid molecule or glycoprotein molecule. So they don't span the membrane, but they are attached to the component of the membrane. So, and here also we can see the cytoplasm or the cytosol, and we can see also some of the cytoskeletal proteins that we'll discuss them later on. So now let's talk about the plasma membrane protein and then move to the other organelles and then the nucleus. The cell membrane or the plasma membrane is a selectively permeable membrane. What do you mean by selectively permeable? It allows certain molecules to pass and other molecules not to pass, and we'll see later on what is the difference. What is the composition of the cell membrane? As I said, it is a double layer of phospholipids due to the hydrophilic parts of the phospholipid molecules, which are the main component of the cell membrane. This phospholipid bilayer restricts the passage of water and water-soluble ions, and also the water-soluble molecules, most of the water-soluble molecules. Another component of the cell membrane are the proteins that either fully or partially span the membrane, the cell membrane, and we call them integral membrane proteins. And we have the peripheral proteins that are attached to the surface. What is the function of the integral membrane protein? They provide structural support to the membrane as well as to the cell. They provide molecules that allow the transport of other molecules or ions, as we'll see in the lecture about the transport across the cell membrane. And also they serve as receptors for different chemicals like hormones and neurotransmitters that need to send a message to the cell, but they cannot cross the cell membrane, so they attach to the receptors present as part of the cell membrane. There are other functions of the membrane proteins, and I will cover them in a different lecture. Also, the cell membrane has negatively charged carbohydrates that are attached to the surface, mainly to the outer surface, either to the lipids, and we call them glycolipids, or to the proteins, and then they become glycoproteins. Now you can see here in this slide we have the nucleus and the cytoplasm, and the cytoplasm is full of different organelles that I'm going to cover in the coming slides. So the cytoplasm is composed of cytosol, which is the aqueous or the watery content of the cell. This is composed mainly of water plus the other ions and solutes. And then we have in the cytoplasm organelles. These are subcellular structures or components that are present in the cytoplasm. Then we have the nucleus, which is the largest organelle in the body. It is a large spheroid body. It is, as I said, the largest organelle present inside a living cell. It contains the genetic material, mainly in the form of DNA. And it has also a smaller organelle called the nucleus. It is the center for the production of ribosomes. And the ribosomes are very important for the synthesis of the proteins that will be used either inside the cell or will be released from the cell, as we'll see later on. As you can see here, we have phospholipid bilayer membrane proteins. We have the peripheral proteins attached to the interior surface of the cell membrane. We have rough endoplasmic reticulum. We have the Golgi apparatus. We have the mitochondria, and you can see very well that the cytoskeletal proteins are present inside the cytoplasm as part of the cytoplasm, and they have very important function. They act as a skeleton for the cell, as a scaffold where organelles are suspended in position inside the cytoplasm. You can see we have different types of cytoskeletal proteins. We have the small ones or the thin ones, we call them microfilaments. We have larger ones, we call them microtubules, and we have those that have a size intermediate between the microtubules and the microfilaments, and we call them the intermediate filaments. So they are very important. As I said, they form a scaffold inside the cell, and one of the functions of the cytoskeletal proteins is to hold the organelles in position inside the cytoplasm. The cytoplasm is jelly-like matrix within the cell. It includes organelles and cytosol. It is highly organized structure with microtubules, microfilaments, and intermediate filaments that function as cytoskeleton. The cytoskeleton is composed of actin and myosin, the microfilaments, and we'll see these two terms again in the muscle tissue. The spindle apparatus composed of microtubules, and this is very important during the cell division, meiosis and mitosis. The cytoskeletal proteins give the cell structural support and enable some degree of motility or contractility of the cells. In the muscle cells, the microfilaments are present in large amount. That's why the muscle cells can contract. Organelles are suspended in the cytosol by the filaments or by the cytoskeletal proteins. Now let's start to talk about the different organelles, and let's start with the lysosomes. What is the main function of the lysosomes? They act as digestive organelles that are present inside the cell. So they are just like the digestive system of a cell. We have different types of lysosomes. We have the primary lysosomes that contain only digestive enzymes. So they contain digestive enzymes and they are ready to digest any vesicle that they fuse with, like during phagocytosis. We have the secondary lysosomes. When they are formed when the primary lysosome fuses with the food vacuole or the organelle. Organelles in the cell, they age, so we need to replace them. So the cell should get rid of them, and this is done by the lysosomes, the digestive enzymes present in the lysosomes. Secondary lysosomes contain partially digestive remnants of other organelles and organic material. And then we can see that also here we have the primary lysosome, secondary lysosome here in the figure, and what is left, or what cannot be digested by the digestive enzymes that are present in the lysosome, will be stored in a very small vesicle on one side of the cell, and this is called the residual body. It contains undigested waste products because the enzymes that are present in the lysosome cannot digest everything. Here we can see the different functions of a lysosome. They can help in the digestion of food, they digest organelles, and also even they digest aged cells from the interior. They release their content inside the cytoplasm, and this will start the digestion of the cell itself. And this is the process called the process of digesting the organelles is called autophagy, the process that destroys worn out organelles so that they can be continuously replaced. So the cell can recycle its organelles and replace them by new one with a more optimal function. There is another term called apoptosis, which is programmed cell death, where the lysosomes, as I said earlier, they release their contents directly into the cytoplasm. So they release their content into the cell, and this will digest the cell from the inside. Another organ is called peroxisome, and plural peroxisomes. These are small membrane-bound organelles present in the cytoplasm, and they have a function a little bit different than lysosomes. They are important for the oxidation of amino acids, fatty acids, and toxic foreign molecules by their enzymes. So peroxisomes are important function of the liver and kidney cells, and this process generates hydrogen peroxide. So peroxisomes are mainly present in kidney and liver cells, and during the oxidation of amino acids, fatty acids, and the toxic foreign material, the free or the hydrogen peroxide is released as a product of these reactions. Peroxisomes, to get rid of the hydrogen peroxide, they contain a reducing enzyme called catalase, because hydrogen peroxide is toxic to the cell, so it should get rid of it, and this is done by this enzyme, catalase enzyme, that will convert hydrogen peroxide to water and oxygen. Another major function of the peroxisome is the breakdown of very long chain fatty acids. We have long chain fatty acids and we have short chain fatty acids. Long chain fatty acids need the activity of peroxisomes to be broken down or digested into smaller fragments. The mitochondria, and this singular is mitochondria, these are the sites for energy production of the cell. So they are the energy factory of all cells that can perform aerobic metabolism. As you can see, each mitochondria has an outer membrane and an inner folded membrane. The folds are called the cristae. And there is an intermembrane space, and you study in biochemistry that the enzymes responsible for the cellular respiration are present in the inner membrane. The mitochondria, they are different from other organelles in that they have their own DNA, so they can reproduce without being dependent on the nucleus. As I said, they have an outer smooth membrane and an inner membrane that is folded, and we call the folds cristae. The cristae and the matrix, or the interior of the mitochondria, compartmentalize the mitochondrial space into different compartments, and these have different roles in the process of energy production or energy generation. Ribosomes, as I said at the beginning of the lecture, ribosomes are protein factories. Wherever they are present, they are involved in the production of proteins, either proteins that will be used inside the cell, or proteins that will be released from the cell, because many hormones and neurotransmitters as well as other signaling molecules are proteins that are produced inside the cell and then released upon the stimulation of that cell. Proteins produced according to genetic information contained in messenger RNA. So the ribosomes will read the code on the messenger RNA and will translate that code into an amino acid sequence, which will form polypeptide and then the protein. Ribosomes located in the cytoplasm free, we call them free ribosomes, or are present on the surface of the endoplasmic reticulum, and if they are present on the surface of an endoplasmic reticulum, we call that endoplasmic reticulum as rough endoplasmic reticulum, to differentiate it from the smooth endoplasmic reticulum, which lacks ribosomes from their surface. Each ribosome has two subunits composed of ribosomal RNA and protein. So as you can see here, this is a ribosome. It has a small subunit and a large subunit. The ribosomal RNA molecules serve as enzymes Also, we call them ribozymes that are required for the protein synthesis. Now, the other organelle is the endoplasmic reticulum, or ER. So as you can see here, we have endoplasmic reticulum. Usually the endoplasmic reticulum is close to the nucleus, especially the endoplasmic reticulum that has ribosomes on its surface. And as I said, we call the rough endoplasmic reticulum. And we have another subtype of endoplasmic reticulum called the smooth endoplasmic reticulum because it lacks ribosomes. And to attract your attention, in some tissues or in some cells, the endoplasmic reticulum, especially the smooth endoplasmic reticulum, has a specific name to that cell. For example, the smooth endoplasmic reticulum in skeletal muscle fibers and cardiac muscle fibers is called the sarcoplasmic reticulum. And it has a very important function in storing calcium that is important for the contraction of the muscle cell. So as you can see here again, this is the nucleus, and I said that the rough endoplasmic reticulum is very closely associated with the nucleus. So we have rough endoplasmic reticulum, smooth endoplasmic reticulum, and you see here the smooth endoplasmic reticulum do not have any reticula do not have any ribosomes on their membranes. The rough endoplasmic reticulum bears or carries ribosomes on their surface. They are present in all cells that are active in protein synthesis. Proteins that enter the cavity or the cisternae of the rough endoplasmic reticulum are modified for secretion. So please remember that most of the proteins that are produced by ribosomes on the rough endoplasmic reticulum are proteins that will be released from the cells. So secretory proteins. The smooth endoplasmic reticulum, its main function is the storage of calcium. And as I said, especially in the striated muscles, and the striated muscles are the skeletal muscles. These are cells present in the skeletal muscles that move the skeleton, and the cardiac muscles that are present in the wall of the heart. They also provide sites for enzyme reaction in steroid hormone production and inactivation. So they are also important for the production and inactivation of one type of hormones, and these are the steroid hormones that are produced from cholesterol. The question is, if we know that rough endoplasmic reticulum is the site for protein production and these proteins will be released from the cell, what about proteins that are used inside the cell as enzymes or proteins that have other functions inside the cell? These proteins are not produced by ribosomes on the rough endoplasmic reticulum. They are produced by the free ribosomes. So we don't need to produce them in the rough endoplasmic reticulum. Proteins that are their fate is to stay inside the cell are produced on the free ribosomes that are present in the cytoplasm. Another organelle, which is very important, it is called the Golgi apparatus or Golgi complex. So it is composed of stacks of cisternae, group of cisternae, between the endoplasmic reticulum and the cell membrane. And from its position, maybe you can guess the function of the Golgi apparatus. The Golgi apparatus will receive vesicles containing proteins that were produced in the rough endoplasmic reticulum. So mainly it is for the final modification of molecules that are produced in the rough endoplasmic reticulum in preparation for their secretion to the exterior of the cell by exocytosis, as you can see here. So Golgi apparatus or Golgi complex is composed of stacks of hollow flattened sacs called cisternae. One side of the Golgi apparatus faces the site for entry of vesicles from the endoplasmic reticulum that contains cellular products. So one side is towards the rough endoplasmic reticulum, the other side is towards the plasma membrane. The other side, side, yes, of the Golgi apparatus faces towards the plasma membrane and releases vesicles of chemically modified products. So the Golgi apparatus modifies or does the final modification of those produced molecules in preparation for their release. So the main function of the Golgi complex is to modify proteins, separate them according to their destination, and then pack them into vesicles in preparation for their exocytosis. Centrioles, we have two cylindrical bundles of protein filaments that are perpendicular to each other. They are present near the nucleus, and they are very important during cell division. They direct the mitotic spindle development during the cell division, mitosis and meiosis. Then we have other cell organelles called the cilia, flagella, and the microvilli. Cilia, they are tiny hair-like structures that project from the surface of the cell. This is an epithelium. Here we have the basement membrane, and the apical membrane of these epithelial cells have cilia that beat in one direction. So they help in propelling fluid or secretions from one side to the other. Say they stroke in unison, so they beat in one direction all together in harmony. Example, the respiratory tract and the uterine tube. So in the respiratory tract, they propel mucus and trapped pathogens and dust away from the alveoli towards the pharynx, so we can remove that mucus in the sputum or swallow it. In the uterine tube, the cilia will help in the propulsion of the ovum or the zygote, and we will study that in the female reproductive system. Then we have the flagella. The flagella, they are simple whip-like structures that propel the sperm through its environment. This is a sperm or spermatozoon. It has a flagellum that beats and helps the sperm to reach the ovum and fertilize it in the ovarian duct or in the uterine tube. And finally, we have the microvilli. You can see here, this is an epithelial cell. It has a cell membrane. The apical membrane, it is thrown into finger or small finger-like projections. They are very tiny. We cannot see them by our naked eye, so we need to see them by the microscope. That's why we call them the microvilli. There are numerous folds, finger-like projections, and the main aim of the microvilli is not motility, not movement like the cilia or the flagella. It is to increase the surface area, and this is very important in organs where absorption is important, like the alimentary canal, especially the small intestine. So they aid in the absorption. Now let's talk about the last organelle or the last part of the cell, which is the largest organelle inside the cell. As you can see in this slide, the nucleus has a double-layered nuclear envelope, an outer layer and an inner layer, and these layers fuse together at the nuclear pore, which is very important for the transport of proteins and RNA between the nucleus and the cytoplasm. We have the genetic material in the form of chromatin present throughout the nucleus, and then we can see this purple organelle inside the nucleus, which is the nucleolus. It is very important for the production of ribosomal RNA, as most cells have a single nucleus later on. Although other cells have two or more, we can see, for example, in the skeletal muscle fibers, we have many nuclei, and this is due to the fusion of many stem cells during the development of the skeletal muscles. So the skeletal muscle fibers are multinucleated. The nucleus contains the genes in the form of DNA that code for the production of messenger RNA. The messenger RNA will be read by the ribosomes to produce different proteins. The nucleus is enclosed by inner and outer membranes, and this is called the nuclear envelope. So the nuclear envelope has two layers, and the two layers fused with each other at the nuclear pores. The outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. The nuclear pores, or the nuclear pore complexes, fuse the inner and outer membrane together. This will allow selective movement of proteins and RNA between the nucleus and the cytoplasm, as I mentioned earlier. The nucleus is a dark oval structure inside the nucleus and is involved in the production of ribosomal RNA. So it is very important for the production of ribosomes, either the ribosomes that are attached to the surface of the rough endoplasmic reticulum or the free ribosomes that are present in the cytoplasm. As I said, that the nucleus contains chromatin, which is the genetic material. As you can see here, chromatin is composed of double helical DNA molecules that are negatively charged, and they are wrapped around the histones. These are histone proteins. They are basic, they are positively charged. So because they are positively charged and the DNA is negatively charged, so they form spools around which the DNA molecule or the double helical DNA will wrap. And finally we can see that genetic material in the form of chromosomes that are ready for division, and chromosomes are seen clearly just before the division, and each chromosome is composed of two arms that are connected by the centromere. So the DNA within the nucleus combines with proteins called histones to form chromatin. Thread-like material that makes up the chromosomes. This is the chromatin. Histone proteins are positively charged and form spools around which the negatively charged DNA strands will wrap. And there is something called eochromatin. It is active in genetic transcription. So this eochromatin will be transcribed to RNA, and then the RNA translated into DNA. And there is another type of chromatin called heterochromatin. So the genes in the heterochromatin are permanently dormant. They will be never transcribed, and they will remain inactive throughout the life of the cell. Here in this table we can see the summary of all the parts of the cell: the plasma membrane or cell membrane, the nucleus, and then the cytosol with all the organelles in the cytosol. As you can see, the plasma membrane is composed of phospholipid bilayer with scattered proteins and cholesterol molecules. Main function is to maintain boundary of the cell and the integrity of the cell structure, and we have many proteins embedded in these phospholipid bilayer, and they have multiple functions. The nucleus is separated from the cytoplasm by the double-layered nuclear envelope. It is where DNA is present that dictates cellular function and protein synthesis. The nucleus is important for the synthesis of ribosomal RNA. Then we have membranous organelles. So these are organelles that have membranes, and non-membranous organelles. The membranous organelles mainly are the rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi apparatus, the mitochondria, lysosomes, and peroxisomes. Non-membranous organelles, please remember the ribosomes and the centrioles. And don't forget the cytoskeletal proteins that are very important in providing structural support to the cell, and they help in the cell movement and contraction. With this slide, I end this lecture. Thank you for your patience, and I hope that you enjoyed this session. and you came out with many new concepts.